So today we have Chris Gentile. Chris is a faculty in food science and human nutrition here at CSU. Chris has a pretty fascinating path to coming here, and I just wanted to highlight a few things. He originally got his bachelor's degree from Skidmore, a very prestigious liberal arts school. Went on to a lab that many of us know, Doug Seal's group at UC Boulder. Went out to do his Ph.D. at Virginia Tech. Did his postdoc here with Mike Pagliassati in food science and human nutrition, and then transitioned to a faculty member in the same department. And so what I thought was interesting is Chris is able to grab really pretty much every training award you could get as a postdoc or early career faculty. He was able to get a T32 first, an institutional postdoc fellowship, got an F32, an individual postdoc fellowship, got a KO1, which is the transition to faculty award, and then received the Betcher Award for early career biomedical research, which is a Colorado-specific award. So really awesome. He's published 46 publications to date. He was recently awarded an R01, so he is definitely now in the money. Also has an R21 with our own Frank Dineno, as well as an Innovative Project Award from the American Heart Association. So Dr. Gentile has done exceptionally well as an early, now mid-stage faculty, I guess you could say. And today he's here to share some of the findings from his recent work entitled Exploring the Causes of Obesity-Related Vascular Dysfunction. So thank you for coming.
All right, thanks for coming, everybody. There's a lot of people here. It's almost as if some of you were bribed. Half of these are my students in 350. They were actually bribed. So. Okay, so what I thought I would do is basically just give an overview of the lab and what we've done over the last 7 or 8 years. And I thought I'd start, I'm getting a little too old to start this way, but I thought I'd start with a little bit of my training, just because what I do now actually was resulted directly from my training. So I received, like Dan said, I received my master's degree in Boulder in the Department of Integrative Physiology, and I then went to Virginia Tech and did my Ph.D. with one of Doug's former postdocs named Kevin Davey. And what we did, research-wise, when we were there, is essentially this. We looked at how obesity and diet affected cardiovascular health and how that sort of informed cardiovascular disease risk. And it was all clinical, so we did it entirely in humans. And then after 2006, I left there and I came here. And I worked specifically with Mike Pagliassati, who's now the department head in nutrition. And I went there to try to get some experience with models, cell models and animal models, because everything we had done was with humans. So I joined there in 2007, and in 2011, I transitioned into a faculty position. So when I was starting my own lab here, we essentially tried to follow the same model, right? We wanted to look at obesity and diet and how it affected cardiovascular health and disease risk. But importantly, what we wanted to do was take these model systems from my postdoc, right? So we wanted to do this in cells, animals, and humans as well. And when it comes to cardiovascular health, there's a whole bunch of things we can look at, right? That's a pretty broad term. I just threw up some random ones here. We can measure blood pressure, cardiac structure and function. But really, in my lab, what we look at is blood vessel function. And more specifically, we look at 2 parameters of blood vessel function, endothelial function and arterial stiffness. So this is sort of a general overview of what the lab does, and I'll just take a few minutes. I know a lot of you know this very well, what endothelial function and arterial stiffness are. I'll just give you a quick background of what these 2 do. Okay, so I'll start with endothelial function. So the vascular endothelium is here, right? It forms the innermost layer of a blood vessel. So it's in this strategic interface that sees the blood flowing through the vessel, and it can relay information from changes in the blood to the underlying smooth muscle. Right? So the endothelium and the smooth muscle cells can work jointly to adapt to changes in the blood vessel, and then they can constrict or dilate as necessary to whatever the stimulus is. So the endothelium, that underlying in a blood vessel, is really important to cardiovascular health, right? Overall cardiovascular homeostasis. So for instance, it can produce and secrete a number of different factors that are anti-inflammatory, thrombolytic, anti-proliferative, vasodilatory, right? So a healthy endothelium is indicative of overall cardiovascular homeostasis. Now, when the endothelium becomes dysfunctional in these states that you're all familiar with, right? Smoking, obesity, aging, sedentary lifestyle. When those physiological states are present, then this endothelium can be dysfunctional, right? And when that occurs, then the profile of the endothelium becomes more pro-inflammatory, more thrombotic, proliferative, and can induce vasoconstriction. And if these things all occur, then we can have disrupted cardiovascular homeostasis, right? So this endothelium plays a large role in overall cardiovascular homeostasis. And we can measure this, right? We can measure this in humans. This isn't directly what our lab does. Frank and others do this. But essentially in humans, we focus more on the vasodilatory capacity of the endothelium. So really briefly, there are other ways, but there are two ways here that we can measure this. So essentially what we're doing, this is flow-metered dilation on top. And what this entails is taking a blood pressure cuff and tying it around somebody's forearm, and you can occlude blood flow, so pump it really high, blood flow is occluded, and then you let it go. And what that does is cause an increase in blood flow through the forearm. Right? And then you can use an ultrasound and actually measure the change in diameter of that blood vessel. Right? So before the cuff is released and after. And the larger the increase in vessel diameter, the greater the endothelial function. Okay? We got it. And then here we do the same thing. We're just measuring blood flow. We're not measuring it with an ultrasound. We're measuring it with a plethysmograph. But essentially we're doing the same thing. The main difference in this technique is we're not inducing a physiological increase in blood flow. So it's not occlusion, we're actually infusing drugs. So acetylcholine is a drug that actually causes the vessel to dilate, right? So the same thing is, essentially the same thing in both scenarios. One is just a physiological inducer of blood flow, one is a pharmacological inducer. We can do this too in, before we get to the animals. So the reason why we're interested in this is because if you use either one of those techniques, an increase in blood flow, or that is an increase in endothelial function, is associated with less of a chance of cardiovascular events, cardiovascular disease, cardiovascular death. So this is one graph of many that I just pulled off from a study just to illustrate the point. So here is survival, right here, and this is in about 600 subjects, and they followed these subjects for about 4.5 years and measured endothelial function. And so what you can see is that individuals who dilated greater, with greater endothelial function, they had much greater survival. If you didn't dilate as well, you had less endothelial function, then you had much greater risk of death. And like I said, there are dozens of studies that corroborate these. Okay, so in my lab we do this, but we don't do it in humans. We actually do it in mice, and we have done it in rats. The technique, though, is pretty similar. So we take a mouse. What's a little easier with a mouse is we can actually pull out the blood vessel, right? We don't have to image it, we can actually pull it out of the mouse. So we extract the blood vessel and we put it on these little glass cannulas. And so here is the blood vessel here. It sits in sort of this warm physiological bath. It's happy, you can dilate and constrict even if it's out of the animal. And we tie it off, and then we put in these drugs just like with plethysmography. And so here is, here's a vessel. That's actually a mesenteric artery of a mouse. So what we do is we drop on a drug that causes constriction first. So this is the vessel in real time. We just dropped on a drug that's going to cause constriction, so you can actually see and watch it constrict. Which is always kind of cool to watch, right? You can just see it constricting. I still think it's cool. And then once we do that and cause constriction, then we can put on our dilators, right? And so here's our dilator, acetylcholine. And so what you can see is with increasing doses of acetylcholine, here's our curve. We have a dilation curve. And so what we'd see in something like an obese or an older individual is essentially just dilate less, right? So mice behave just like the humans do. Okay, so we'll see a lot of graphs like this. So that's how to look at it. Essentially, this is the rate of dilation. Okay, so that was endothelial function. That's one of our two main outcomes that we use in the lab. The second one is called arterial stiffness. Sorry, this slide doesn't look like it should. Sorry, I'm looking at this. This isn't the right slide. Obviously not right. Sorry, let me see something there. You know what? That wasn't the right presentation. Sorry, let me just open this for a second.
I'm down back in there. That's for his
Okay,
other HES talk.
This is what I should have. Sorry about that. I recovered okay. Okay, so this is the second technique we use in my lab. So this is arterial stiffness. So when the heart beats, it's shown here. So here's a beating heart. The aorta, that large vessel coming off from the heart, is going to dilate, it's going to expand, and then it's going to contract, right? And it does this in part just to accommodate the increase in blood flow that's coming from the heart, right? So here's a healthy heart, you see it expands, and then it contracts. And here's an unhealthy artery, right? So this can be induced by the same physiological, pathological states that I mentioned, aging, obesity, smoking. This vessel doesn't expand and contract as well, right? Because it acts more almost like a metal pipe. It's much stiffer and can't expand, okay? Now, this stiffness is what we can measure in the lab. Now, when the heart beats, there's a pulse wave with every beat. So this happens with all of our heartbeats. There's a pulse wave, kind of like a sound wave, that's generated with each beat of the heart. And this pulse wave travels through the arterial tree. When you have a state like obesity or aging, where that vessel is stiffer, then this pulse wave travels much faster, and that should be moving very fast. It's not moving. But trust me, there's an animation. What it shows is that this moves much faster. Okay, so that pulse wave, when you have a stiff vessel, that pulse wave is going to travel down the arterial tree much faster. Okay, and so we can measure this. So we measure this with a technique called pulse wave velocity. Right, so we're measuring the velocity of the pulse wave. We can do it in humans, so we put essentially 2 probes on 2 spots of a human, or we can do the same exact thing with a mouse. So we put 2 probes, the mouse is just anesthetized, and then we put 2 probes, and we essentially look at the speed of this pulse wave as it travels down the arterial tree. So here is the waveform that's actually generated for this pulse Doppler system. So you can see here's one waveform from the probe, and here's the second one, and you can see this is offset, so this starts a little later, right? So we're essentially looking at the distance between when those 2 pulse waves start. And then we simply measure the distance and the change of time, and we have pulse wave velocity. Okay? And again, this is what we use as an indicator of arterial stiffness. Now we do this because just like endoplasmic function, pulse wave velocity is very closely related to cardiovascular events and mortality. So this is another study, again, there's several like this. They followed about 2,200 individuals over 8 years, measured their pulse wave, and then measured what happened to them over those 8 years. And you can see people who have higher pulse wave velocity have a much greater risk of cumulative cardiovascular events. So here what they did is they actually took those people, they separated them into quartiles. Individuals with the highest quartile, that is the highest pulse wave, had the greatest risk of events over an 8-year period. Okay? So coming back to here, this is essentially what we measure in the lab. We measure these 2 parameters of cardiovascular health, and we do it with these 3 models in cells, mouse and humans. And really in this schematic, we sort of focus here. So we're not really too interested in this part of it. We don't really look at if somebody develops endothelial function, what's their cardiovascular disease risk. We leave that to other people. We look more at when somebody becomes obese, why do they develop endothelial dysfunction? Or why do their vessels get stiffer? And over the last 7 or 8 years, there's essentially been 2 potential mechanisms that we've looked at. One is endoplasmic reticulum stress, and then the other one is the gut microbiota. Okay, so for the rest of the time, I'm going to talk just about these 2 and show you some of our data showing these. I'm going to start with ER stress. Okay, so ER stands for endoplasmic reticulum. As you all probably remember from your intro to physiology course, the endoplasmic reticulum is essentially an organelle that extends from the nucleus to the cell membrane. It has a whole bunch of functions that I'm not going to talk about. The one function that we are concerned with in the lab is this role of folding proteins. So when a new protein is created, it's the role of the ER to fold this protein into its correct 3-dimensional configuration. And then that protein is functional and can be shipped out of the organelle. So in that regard, then we can sort of look at ER homeostasis as a balance between the proteins that need to be folded and the protein folding capacity of the organelle. Right? If the protein folding capacity is equal to the proteins that need to be folded, then we have balance. Right? When these are imbalanced, and that is the proteins that need to be folded exceed the folding capacity, then we have this state called ER stress. Okay, and this is sort of what we study in the lab as the state of ER stress. When that occurs, ER stress, the ER has this highly conserved compensatory mechanism to try to restore homeostasis, and this is called the unfolded protein response. So really briefly, the way this works, here's a picture of the organelle. Here are some unfolded proteins. You have these 3 proteins that are on the membrane of the ER, IRE1, PERK, and ATF6. These are held in this sort of quiescent or inactive state by another protein called GRP78. When unfolded proteins accumulate like that, and what happens is GRP78 recognizes these proteins, leaves the 3 other proteins, and so now these become activated by GRP78 leaving them. When they become activated, they essentially induce 3 independent but very cooperative cellular signaling pathways. And I didn't put the whole pathways here, but we just have these arrows. Essentially, the point of these being activated is to restore homeostasis. The way it does that is by 1, increasing transcription of proteins that help in folding. 2, it just decreases general translation of proteins. And then if necessary, it causes cell death. So if you think about this, it makes sense, right? If the whole problem is that we have too many proteins and we can't fold them, then one thing the EPR does is just stop making proteins, right? And then the second thing it does is increase the machinery to fold the proteins that are already there, okay? So that's what the EPR does. Hopefully, if that's the case, then we restore ER homeostasis. Okay, so that's the ER stress and that's the EPR. Now, one thing that's relevant just to studying this is that we actually can't measure these unfolded proteins. So when we measure ER stress, we want to look at it experimentally. What we do is we actually measure these proteins with EPR. Okay? And so this pathway, the EPR, became kind of a hot area of science back in about 2004, and has been related to a whole bunch of disease processes, everything from aging to, in our case, we look at obesity. So when I was first starting the lab out, and this is, man, like 8 years ago, this was what we wanted to look at. We wanted to look at the role of ER stress in endothelial and dysfunction and arterial stiffness. But we wanted to just start out really, really simply. So we just started here with some cell models. So we just had some endothelial cells, and at this point, this is 2009-ish, we just wanted to know, is ER stress even present in an endothelial cell? Nobody had really even looked at that, right? So if you cause dysfunction in an endothelial cell, do we get ER stress in there? So we started really simply. We took some endothelial cells, we took a toxin that is known to induce ER stress called tunicmycin, and we just wanted to see if we put this in some cells, do we get ER stress? And so that's what happened. We measured these 3 genes or proteins here, XBP1, ATF4, and CHOP. These are all just part of the EPR. You can see we put in tunicmycin, and gene expression goes up. So at least really simply showing, yes, an endothelial cell does have the capacity to increase the EPR. Then we didn't want to use tunicmycin, right? This is kind of a sledgehammer approach, kind of a toxin. So we wanted to use something a little more physiological, so we used palmitate, right? A 16-carbon saturated fat that sort of gives saturated fats that name. We get put palmitate in the cells, and the same thing happens. So if the cell is overloaded with a lipid, then it too causes ER stress. So from there, we went on, we did a whole bunch of these studies that I won't show you. We used different inducers and time and dose responses. Suffice to say that essentially ER stress is definitely an endothelial cell, and we could show it with things that are physiological like lipids. So then what we did is we wanted to show, okay, ER stress is there, but does it really matter for the cell? So we started to measure things like cell death, right? So this is a measure of cell viability. So this is essentially the health of the cell. The greater the viability, the less cell death, right? So here's a control cell. If we put in tunicmycin and we put in palmitate, then the cell viability goes down, indicating there's more cell death. And then what we used was a substance called TUCA, and TUCA is an inhibitor of ER stress. So what we wanted to show now was, okay, we know there's ER stress in the cell. What if we inhibit that ER stress? Can we actually decrease cell death? So when we do it with tunicmycin, you can see there's an increase in cell viability. Interestingly, when we give TUCA to a cell that palmitate is called cell death, it actually never improves it. And we have some ideas as to why, but TUCA never improves palmitate-mediated cell death hormones. But still, this was concept enough that we do think there's ER stress and it's playing some role, at least in some forms of cell death. And again, we had a whole bunch of other studies kind of corroborating that. Okay, so that was sort of the first work we ever did when the lab was started. And then we wanted to take this to an animal model. right? We showed it in cells, we wanted to now take it to some in vivo model. So what we did to start was we took this endothelial function test that we showed you, right? We put drugs right in the bath from a vessel that's been isolated. And first we started that same drug, tunicamycin. This is the one that increases urinary stress. And we wanted to look at these dilation curves, right? So here's our normal mouse with nothing in there. You can see it dilates this much, about 80%. When we put tunicamycin in, it decreases to about 45%. So showing that when you give an ER stress inducer, the vessel becomes dysfunctional. And then we gave TUCA to see if we could improve it. And you can see there was at least a partial restoration of function. Okay, so if we give ER stress, it reduces endothelial function. If we give TUCA and inhibit ER stress, then it restores some of that function. So again, kind of making our way towards a more kind of realistic physiological model. Then we took two mice. This is a control mouse, and this is a DB mouse. So DB mouse is lacking a leptin receptor. It's very diabetic, it's very obese, and we know that it has endothelial dysfunction. So what we wanted to do first was to just put TUCA in this bath. So if we have this mouse that causes dysfunction, can we improve it with TUCA? And so here are the data. So you can see this diabetic mouse, it's pretty incredible. It just doesn't dilate at all, right? That's a pretty dysfunctional vessel. Here's a control mouse, and then here is our TUCA DB mouse. So you can see again, functions restored by about 40 or 50%. Okay, so that was good enough. We had done that, although the one limitation was we were still just putting this drug right in the bath, right? I mean, we still weren't actually getting anything in vivo. So then we decided finally to start a study. I think there's enough evidence. We can actually just try this in an intact mouse. So we decided to give TUCA to these DB mice, but we gave it IP. So we just injected TUCA and then let them go for 4 weeks and see what happened. So here are the data for this. So you can see here's a diabetic mouse skin. Here is our dilation of a normal mouse. And you can see that TUCA given in vivo essentially looks exactly the same as given acutely, right? It restores function by about 40%. And then since it was in vivo, we also could use our other type of vascular dysfunction measure. This was pulse rate velocity. So you can see here's pulse rate velocity of the control mouse. The DB mouse is really high. And again, this pulse rate is going faster, so that means that the vessels are stiffer. When we give TUCA, we can see that that pulse rate velocity is reduced. Okay? So that was pretty good evidence, at least in the mouse model, we thought, that that ER stress is playing some role in vascular dysfunction. The goal of this obviously isn't to try to help vascular dysfunction in a mouse. It's to take this to a human, right? So what we did next was we took this to a human model. At the time, this was several years ago, there was some evidence that ER stress was present in humans that were obese in various tissues. So for instance, we knew that there was liver could express ER stress, the adipocytes, and the pancreas. Nobody really had any good idea of whether or not ER stress was present in the vessels of an obese human. Now part of this, so with that said, the goal, just the question we asked was, do obese individuals with endothelial dysfunction display markers of ER stress in vascular endothelial cells? Right? So that was kind of our next question. And part of the reason why this hadn't been done is because if you think about it, it's easy in our mouse models to just pull out a vessel and measure something in that endothelial cell. It's pretty hard to do that in human. So we collaborated with Doug Seals in Boulder for this, and so they have a technique where they can actually take this little, what's called a J-wire. So when they're putting a catheter into a human, you can use this little kind of curved J-wire and actually scrape the side of the blood vessel, be it a vein or an artery. Some of those endothelial cells actually rub off on the J-wire. You can take those cells and process them and then measure proteins from there. Right? So we use this technique from Doug's lab, and we used it on these obese individuals to see if those with endothelial dysfunction displayed markers of ER stress. So here's some of the data. There's some non-obese and obese individuals, just some characteristics. You can see they were in their 50s. By design, they were obese, they had a higher waist circumference, and higher triglycerides. And here's our endothelial function data. So you can see the obese individuals dilated less, right? And again, this was the whole purpose. We were hoping they had endothelial dysfunction. The question was, did they have ER stress? And they did, at least for the proteins we looked at. So we looked at these 3 proteins that were those kind of initiators of the cell cascade, right? Those transmembrane proteins, ATF6, PERK, and IRE1. And you can see in the obese individuals, in every case, this ER stress is present. Okay? So this was really the first evidence that there was some ER stress, at least in the blood vessel of an obese individual. So where we are now, you're not looking, Frank. So what we're doing now is we're going to actually look to see if TUDCA can restore endothelial function and vascular dysfunction in general in obese and older individuals. So again, we don't do this in humans, so we're collaborating with Frank Dano's lab, and they're going to do most of the measures. Nate Bachman is running the show on this. So we'll see, we'll know in 6 months or so whether or not TUDCA is actually viable in humans to improve vascular dysfunction. So that's sort of the ER stress story. Took me like 9 years to collect those data and 20 years to summarize that. Took a lot longer to collect. But that's kind of where this stands right now for ER stress. So the second thing that we've been doing and really focusing more on in the last 4 or 5 years is this gut microbiota. So I'm going to transition for the rest of the talk and just talk about that and some of our data. So I won't get too much into background. I assume all of you have heard something about the gut microbiota somewhere. I feel like it's in the news every day. Essentially, these are microbes that they're around us, on us, in us, right? And we tend to focus on the microbes that are within the GI tract, particularly in the colon and the small intestine. So there's trillions of these bacteria that are within our GI tract. We've known this for probably 100 years that these things exist. We've also known that external stimuli can change the composition of those microbes, right? So stimuli as such as the birthing process, where you live, whether you take certain medications, diet, which is most relevant for us, all these things can change the composition of the microbiome. Again, we've known that for a very long time. If you have certain changes in the gut microbiome, some that are pathological, again, that are induced by these states here, you can develop a state that is termed dysbiosis. And some people don't like to use this term, but just to simplify it, dysbiosis is essentially the state where the microbiome is out of homeostasis. Okay, so it potentially is characterized by potentially more pathogenic microbes within your GI tract, and it may cause some dysfunction within the whole body. So again, these things have been known. What we really haven't known until, say, the last 15 years is that if these microbes change within the GI tract, it essentially can affect a whole different roles of physiological function and may play a role in a whole bunch of diseases. Everything from multiple sclerosis, I won't read through these, but a whole host of different chronic diseases. So this is what we do, is essentially look at these changes in the microbes during obesity and whether or not they play a role in vascular dysfunction and cardiovascular disease. So to look at this dysbiosis just in a slightly different way. So here's your healthy microbiota here on the left, and then it kind of can move towards a dysbiotic microbiota given these pathological states. And what that can be characterized by is reduced diversity in the diversity of the actual microbes, inflammation within the GI tract, and then also what's called intestinal permeability. Essentially what this means is that the permeability of the GI tract itself is increased, right? So the GI tract is essentially kind of a barrier between what's happening there and what happens in the rest of the body. If it becomes permeable, then some things can actually move out into the general circulation and may cause harm. So one of those things that can move out and move into the general circulation and may cause harm is something called lipopolysaccharide, or LPS. So this is a component of the cell wall of certain bacteria, gram-negative bacteria. So there's LPS probably in all of our intestines right now. The intestine can handle LPS quite readily. What happens though, if we have a dysbiotic microbiota, is this LPS can actually get into the general circulation. It can get into the general circulation, it can bind to a receptor in various parts of the body called the TLR4 receptor and induce an inflammatory response. Okay? And this is a very strong inflammatory response that's been implicated in things like septic shock and things that are acutely very deadly because the inflammation is so great. Okay, so our studies, that's just sort of a background on the microbiota. I'll come back to that, but our interest in LPS actually started well before I think I even knew what the microbiota was. This was back again when we just started the lab in 2008 or 9. We had these cells going and we were just trying to, again, see what happens when we give certain substances to an endothelial cell. One of the substances we gave was LPS, and we knew that it caused inflammation. So again, we just wanted to see if our cell model was viable, so we put LPS in, and you can see these are just various inflammatory proteins. So this is just early evidence showing if we give LPS to an endothelial cell, we'll get inflammation and the cell will at least be less healthy. Okay, so these are really early data from the lab. Again, just like ER stress, what we did then is we took this into an animal model and we gave LPS directly in this bath. And here are the fetal function data, so you can see here's our normal mouse, and then here's our mouse with LPS, so there's a reduction in dilation. And then we had some mice that lacked this TLR4. These were from Kelly Santangelo in MIT. And so we took these vessels from these mice. Again, TLR4 is the receptor through which LPS acts, right? So the idea is if they don't have TLR4, then LPS shouldn't be detrimental, and we shouldn't have a reduction in dilation. And that's exactly what we saw. So this is at least some evidence that LPS can affect endothelial function, and it does so through the TLR4 receptor. Okay, so we had those data for a long time. We actually didn't really do anything with them. And we were thinking about this more in terms of just septic shock and certainly not the gut microbiota. That changed with a couple of things. One, there was some evidence that started to accumulate that this increase in LPS in the circulation wasn't just happening in these acute, dangerous situations such as sepsis, but it actually was happening in states of obesity. So the idea was that somebody would become obese and just a little bit of LPS would come into the circulation, just enough to cause this chronic low-grade inflammation. And that inflammation over time then could cause diseases like diabetes and heart disease. So there was some evidence that this might actually be relevant in humans, even in things like generalized obesity. The other thing that happened was that we had a new graduate student from the lab named Michael Batson. He had worked in Doug Seal's lab, and he was really, really interested in the microbiome and really wanted to study it in some capacity. And the other thing that happened was Tiffany Weir, who has the office next to me in nutrition. She's an expert in the microbiome. So I generally don't know anything about the microbiome. So usually when I say something about it, I look at her to see if I'm making a fool of myself. She's not here today, so no microbiome questions until later. But she's really the one who knows everything about the microbiome. I mean, I really just kind of pretend. But she knows a lot about it, and so we thought, well, with Micah's interest and with Tiffany's expertise, we probably could do something with the microbiome. And we had all these LPS data, right? So we thought, well, maybe we can test this hypothesis, right? LPS come in, and maybe that inflammation generated from LPS is part of the reason why we're getting vascular dysfunction. So that's sort of what started us on this kind of journey with the microbiome. And so when we were starting out first, we didn't really know what to do, right? How do we actually study this? There's essentially 3 models, mouse models, that you can study the microbiome. One is you can give an antibiotic cocktail. And so what this does is essentially just wipe out the microbes. If you want to know if the microbes are playing a role in some disease, we'll just wipe them out with antibiotics. So you can do that. You can also do a transplant. So you can actually take the microbes that are in the feces of a mouse and transplant them to another mouse. Again, if the microbes are really mediating some disease, and this recipient mouse should actually acquire that disease. Or you can use what's called a germ-free mouse. And so these are mice that don't have any, endogenously don't have any microbes. They've been born and raised in a sterile environment. So you can see that if we don't have any microbes, well, do they develop the same diseases? We can then combine this, and we can actually transplant different microbes into the germ-free to see if their phenotype changes. So these were the 3 options we had. We didn't really have a germ-free facility. We didn't really know how to do transplant studies, so we defaulted and said, okay, let's just try this with antibiotics. So the first study we looked at was essentially a study with antibiotics. We took mice, we gave them just a normal chow or a Western diet. So this is just a high-fat, high-sugar diet that we know causes vascular dysfunction. We've shown that in the lab before. We put them on this for about 5 months, and then in one group, we gave them antibiotics. Again, with the idea being that if the microbes were causing some of the vascular dysfunction, and if we wiped out the microbes, then the vascular function should improve. Right? So these are some of the data. This first slide just shows, here's just our bacterial count. We just wanted to prove, did our antibiotics actually wipe out the microbes, right? And they did. So the actual intervention was successful. And then this is a PCA plot. So the way to read this is essentially looking at how close the different circles are, the proximity of the different circles. So you can see these white circles. These are settler diet-fed mice. So these are just a control mice. They're all clustering here, whereas our Western diet-fed mice are clustering over here. So this is essentially showing separation in the microbiome composition, right? So this microbiome here is much more similar to these. So essentially what that's showing is that our Western diet induced some change in the microbiome, right? Okay, so here is arterial stiffness. This is our pulse rate velocity. So you can see that here's a control mouse over 7 months, and then here's our Western diet-fed mouse, so it's higher, so higher arterial stiffness. When we give an antibiotic, it goes back down to control levels. Whereas if we leave them on, they just keep getting higher. So it did seem to restore or reduce the vascular stiffness. Here's endothelial function. Again, here's our Western diet fed mice. They don't dilate as well. We give them an antibiotic, and it pretty much restores their ability to dilate. And then we measured some things in the tissues and the blood, just to get an idea of what's happening biochemically. So this is LBP. This is essentially, we use it as just a marker of LPS signaling. Specifically, this is a protein that's binding to LPS before it's binding to TLR4. So what we show here with Western diet-fed mice is an increase in LBP, so that's essentially showing that LPS signaling is increased. When we give an antibiotic, it goes back down to normal. IL-6 is inflammatory cytokine, again, up in Western diet and down in antibiotics, to show that the inflammation that was caused by the Western diet is now decreased by the antibiotics. And then we did a whole bunch of other biochemical measures. Those were in the blood. We did some actually within the aorta, because again, that's where our dysfunction is, right, within the vessel. So we did it within the blood vessel, and then we did it in PVAT. And PVAT is just the fat that surrounds the vessels, right? So our blood vessels have fat that surrounds them, and there's some data that that fat can actually influence vascular function. So again, we're just looking at inflammatory markers are up with the Western diet and restored with antibiotics. This again is looking at NADPH oxidase is up and then down. And then these are ages that may participate in stiffness, the same trend, up with Western diet and restored with antibiotics. So overall, this is a pretty good evidence in our mind that at least in this mouse model, that antibiotics, if you wiped out the microbiome, you actually could restore dysfunction that was induced by obesity and Western diet. So we finished that study. We actually used some of the data for preliminary data, and they came back and they said, well, that's all well and good, but we don't really like antibiotics, so we wanted to do something else. So they specifically asked us to do a transplant study. We had to stay away from it because it's hard, and we didn't really know how to do it. But because reviewers asked us to do it, we tried to do it. So what we did next then was we took control mice, and these are OB mice. So these are kind of like the DBs I talked about before, similar. It's a monogenetic form of obesity. They're lacking the protein leptin, and that lack of leptin just causes them to eat a lot and become really obese, right? So again, just like the DB mice, this is our obesity this is our model of obesity that we know should have vascular dysfunction. So here, first what we did is we just want to prove, okay, we need these mice to have dysfunction, so we need to prove it. So let's measure it in the sub-cohort of mice. And so that's what we showed here. So here's dilation in the obese mice, and it's reduced compared to controls. And then if we look at pulsar velocity, you can see it's higher, right? So at least these mice have the vascular phenotype that we were looking for. Then what we did was, the other thing I should mention, one more piece of data. We used this technique called phytidextrin. So this is actually putting in a labeled, a fluorescent dextrin into the mice. We gavage it into the mice. And so what this does is, if the mice have an increase in permeability in their GI tract, right, in their gut permeability, may be caused by dysbiosis, some of that fluorescent phytidextrin will actually go out into their bloodstream. So what we do is we gavage, we put down phytidextrin and gavage, kind of like a little needle down to the throat, and then 4 hours later, we take blood samples and see if the phytidextrin has gotten into the bloodstream. If there's phytidextrin in the bloodstream, it's indicative of permeability in the gut. So you can see here, the obese mice have an increase in gut permeability. Okay, now what we did, now that we established that phenotype, is we transplanted the microbiome. So we took the microbiome from controls and we put it back into controls. So this is just kind of our control group. And then we took it from the obese and put it into obese, and that's another control group, right? And what the real experimental groups though was we took the microbiome from these control mice and put it into the obese mice. And then took the obese mice and put it back into the control mice. Again, with the idea that if the microbes are causing dysfunction, then the phenotype, the vascular phenotype, should be exchanged between these mice. And so how we did this, we gave a really short dose of antibiotics first, just for a couple weeks, just to kind of knock out the microbiome. And then we transplanted, and this is in the recipients, and then we transplanted via gavage for 8 weeks. We did it daily for about 5 days, and bi-weekly boosters is what we did. So that's how we transplanted the microbiome. And then here at the end, we just measured all of our vascular outcomes. So this, again, is this PCA plot. So we're looking at the proximity of all the same colored circles. So if you look here, here's our control. Control is in red, so they're kind of clustering here, showing that their microbiome is pretty similar, whereas the obese are way up here. So they have in purple, so they have a different microbiome. Once we gave the controls the obese microbiome, what you can see is there's sort of this migration upward, right, towards the obese mice, showing that the microbiome of the control mice starts to look a little bit more like the obese mice. And then once we take the obese mice and give them the control, you can see again they sort of shift downwards and their microbiome starts to look a little bit more like the control mice. So this was at least some evidence that maybe our transplant was working. So here's pulse wave velocity. So what happened was in the control mice that received the obese microbiome, pulse wave went up, so their vessels became a little stiffer. In the obese mice that received the controls, it actually didn't change. So we were not able to decrease stiffness in the obese mice. And we have some ideas why, but that's what the data show. And then that matched collagen, right? One of the reasons why vessels can become stiff is because they can have an increase in collagen. And so you can see the control mice that got an obese microbiome had an increase in collagen, but there was no change in the obese mice that received controls. Phytidextrin, you can see here, here's an increase in the control obese again, but no change in the obese mice that received controls, so same trend. The only thing that really changed, at least for these biochemical things, in both directions were short-chain fatty acids. So short-chain fatty acids are derived once the microbes start to metabolize carbohydrates. So they metabolize carbohydrates and short-chain fatty acids are produced as a byproduct. And there's some data showing that these short-chain fatty acids are actually protected, right? So this might be one of the ways that the microbiome is protected. And so that's sort of the pattern our data showed. You can see that here's the control mice, they have more short-chain fatty acids, and then once they get the obese microbiome, it decreases. And then here's the obese, once they receive control, they increase, right? And that's the case with all three of these short-chain fatty acids. So these change in both directions. What we also did too, I forgot to put a picture up of it, but we gave the hearts to Adam Chico. Adam Chico's a professor in physiology. And they have a technique where they actually can take the heart out of these mice. They put them sort of on this rig, and they essentially induce a heart attack via causing ischemia and reperfusion, and look at the extent of cardiac damage during a heart attack. And so you can see here, here's the control mice that receive the obese microbiome. The infarct size, or the cardiac damage caused by the heart attack, is increased. Here's the obese mice. We give them the control microbiome, and the infarct size, or the extent of damage, is actually decreased. So these data worked out too and matched the short-chain fatty acids. The other thing I'll mention is we actually had an endothelial function from this data. You all can ask me about it later. That technique of endothelial dysfunction, although the data look nice and clean up here when I show them, it's actually kind of a pain in the butt. And the last few studies, it hasn't been working. The constriction and dilation hasn't been working that well. So in the lab, one of the main things we're doing in the lab is actually trying to work out what's happening to our model system endothelial function. So we didn't have really clean data from this study, unfortunately, to see what was happening with endothelial function. But yeah, you can ask me that. Okay, so those are the two main studies that we've done so far with the microbiome. We have a current study that we're kind of halfway through, and we decided to use this third model, this germ-free mice. We don't have a germ-free facility. You can imagine if these have to be born and raised in a sterile situation, there's a whole bunch of equipment we need. We don't have it here, so we collaborated with the folks down in Denver, and they have a germ-free facility. And so this is what we sort of asked a question, and it's similar to what we did in the second study. Do germ-free mice acquire the metabolic and vascular phenotypes of lean and obese human donors? So this time we just took lean and obese humans, we took their microbiome, and we're putting it into germ-free mice to see what happens. So that's sort of an illustration of what we're doing. Here are the subject characteristics. So just to show you, these are the humans, so these are our donors. You can see they are obese, they have a higher blood pressure. This is a measure of endothelial function, and that's increased. And they have increased stiffness. So they have the vascular phenotype we want. Now we have to just transplant this into the germ-free mice. So I'm going to show you one slide. I'm a little hesitant to show you this because it makes the story look so good. I don't know if it's a story. But here is the dilation in the mice that received a lean microbiome and obese. So you can see it happened exactly the way we wanted. We had several cohorts, and we don't think the second cohort worked like this, and we're trying to figure out what happened. So the first cohort worked really well. This is still an ongoing process. This is pretty early, and we need to analyze this data over the next several months. But that's one of the studies that we're doing. The other two studies that we are starting up soon is essentially looking at LPS and whether or not LPS is required for some of these changes, right? It might be LPS that doesn't actually play a role in any of this. So the first thing that we're going to do is take these TLR4 knockout mice. Again, these are mice that don't have signaling through that receptor, so LPS shouldn't be harmful. We're going to put them on a Western diet, and what we think will happen is that these mice should develop gut dysbiosis, right? Because the TLR4 receptor, the dysfunction is more distal in the vessels. So they still should develop gut dysbiosis, but they should have preserved metabolic and vascular function. Even on a Western diet, they should be protected because LPS can't signal. So hopefully that happens. If that happens, then we're going to take the microbiome from those mice and transplant them into control mice. So even though these mice have preserved vascular function, their dysbiotic microbiota should cause vascular dysfunction in the control mice. So this is a study we're going to start up in the next few months. We'll see how it goes with this study. One more study with LPS. We have a collaborator at the NIH, Bob Munford, and he identified a protein called AOAH, acyloxyacid hydrolase. This is essentially an enzyme that breaks down LPS in the bloodstream. So if LPS is playing a role, then mice that don't have AOH should be more susceptible. So that's what we're testing. We're looking at whether mice with impaired ability to detoxify LPS are more susceptible to metabolic and vascular damage upon LPS exposure. So we've done one little quick trial just giving them LPS, just injecting it. We're going to do one more, see if they're dysfunctional. If they are, then that'll give us sort of a scientific justification to give them a Western diet and see if they're more susceptible. So those are sort of the studies that we have coming up. And we'll finish those in the next year or so. Okay, that was it. That's all I got. So before I end, let me just thank these people, the lab members. I mean, really, I'm more of a figurehead. I'm not doing much of anything in the lab. So really, these people on the left are the ones who are actually collecting all this data. So thank you to all these people here, our collaborators on the right. And yeah, that's it.
Lots of time.
I explained it so well, but no one.
I start with a question. So you showed with the PCA that you had different gut microbiota in your OB mice compared to your wild types, but they're on the same diet, right? The OBs just ate a ton more. Is there?
And
right?
Yeah, so
I'm just wondering what the explanation for is. You're eating the same diet and they're missing a leptin receptor. Why would there?
Yeah, so there's some data, believe it or not,
change?
there's some data that leptin actually itself affects the microbiome, so that could be it, that they don't have the leptin. And then whether it's that or whether it's just the accumulation of body fat and the changes that occur metabolically.
Kind of like feeding.
Yeah, right, feedback and change the microbiome. I mean, that said, I'd say one of the reasons we probably didn't see is one of our thoughts, why we didn't see some of these changes, for instance, in pulse wave on the obese mice, going back to controls, like here, for instance. It probably wasn't a very good decision on our part to choose an OB model, right? I mean, if you think about that, this model, this is an OB mouse that has a monogenetic form of obesity. It may be that the gut microbiome plays no role at all in that model, right? It plays much more of a role in a Western diet fed mouse. To be honest, we chose this because reviewers asked us to do it. We didn't want to feed mice for 7 months. That was really the reason. We didn't have to feed them at all because they're already obese. In hindsight, it probably wasn't the best model. I think it's reasonable to speculate we would have seen better results in a Western diet.
Oh, correct me if I'm wrong, but I thought I heard you say that collagen here increases the stiffness of the vessels, and how does that differ from marketing collagen for supplementation?
Yeah, that's a good question. I don't really have an answer to. So collagen will definitely, an increase in collagen over, say, the lifespan will definitely increase sort of the stiffness of the vessel. Just decrease the ability, those matrices will decrease the ability of the vessel to expand. So collagen, taking it, when do people take collagen? Anybody have an answer on that?
I just rub it onto my knees directly.
It's used in cosmetic projects because it's supposed to make you look younger.
Yeah, so I think, if you're rubbing it if you're on your skin, that's very different than collagen actually increasing in things like this, right? That's a very different thing than actually rubbing it onto your skin. Howard. Are you really doing it?
No.
The concept is consistent though, right? So the idea of using collagen for your joints is to strengthen them, right? Or the ligaments, right? But you want certain things to be
Rigid.
To be rigid.
Fair enough, yeah.
Yeah, but not
Not
blood
us,
vessels.
yeah. Yeah.
In your ER stress studies, when you took that, when you saw the improved vasodilation,
Yeah.
you never had a control
No.
cluster condition. So,
We didn't.
so I guess what I'm asking is, does TUCCA have any vasodilatory?
Not in our hand. We've actually looked at it in terms of just in a control mouse.
Uh-huh.
Yeah, in our controls, it didn't when we gave it acutely. I don't know if I showed that, but no, it didn't in a control model. But you're right, in the in vivo study, we didn't give it to our control mice, and that was just the reason we didn't do that is because the mice. We just essentially didn't have enough mice and didn't want to order more because the ages would have been different.
Sure.
So we thought, well, let's just do it the way it is, even though we don't have a proper control. I don't think in a control mouse it's changing anything, at least acutely. Yeah. But you're right, we're kind of missing a control group. If you looked, we're actually missing a control group in the antibiotic study too. Same thing. Yeah.
So, just trying to link the two, arms of your research agenda in the lab right now. Have you thought about treating these endothelial cells with LPS or stimulate TRL4 to see if that induces ER stress? Like, are these things linked?
Yeah. So and I'd love to link these two. We've thought about it. We've looked at ER stress markers, for instance, after the antibiotic study, and they didn't change. I think we even looked at them after the transplant study, and they didn't change. So that was some evidence that at least if you change the microbiome, you're not going to have changes in ER stress or separate. The specific study you're talking about, we've never done. Yeah, so we could look at that too. There are some data that suggests that maybe they're linked, but every time we look at it, we never find them.
Just a real quick follow-up. If you give a blood vessel LPS or stimulate the receptors, do they dilate?
If you give
Acutely?
them,
I'm just thinking
Oh, yeah.
the major problem
Yeah.
with sepsis is hypotension and
Right.
not being able to control blood pressure.
yeah.
So I'm wondering if these vessels actually dilate
Yes.
that, even though.
So we have to use U4/6. So we usually use phenylephrine as a constrictant. Phenylephrine, they don't constrict very well. So I think there is a dilatory effect. So we use U4/6 because it's a stronger constrictor. And that's what most people
What is
have.
U4/6?
It's a prostaglandin. Thromboxane. Thromboxane agonist. So I think there is a dilatory effect because they just don't constrict as well. Yeah. Yeah.
What do you think is mediating the improvement in arterial stiffness with TUCCA or antibiotics?
What's mediating the decrease in stiffness? Well, again, maybe it's something structural. Like,
Can you repeat that?
yeah, so the question, if I got it correctly, is what's mediating the decrease in arterial stiffness with any of the interventions? Again, it might be something structural, like collagen. It might be just get some of these things like ages were decreased in the antibiotic study. That's the only real evidence that we have. Whether it's a kind of structural change or biochemical change. Those are the only two things that we found that tend to consistently change. Yeah.
Do you have any hypothesis for how that's linked to, I guess in this case, like the antibiotics or the intervention?
Yeah, not really. Again, all we've done for biochemical stuff is looking at the kind of usual suspects, things like it was inflammatory cytokines that I showed. I'm not sure. From the antibiotic study, we had a lot, right? So like these. So whether or not it's downstream of inflammation, we don't really know. Yeah. I don't have a better answer for it. But to be honest, we haven't really looked. It's not really been on our radar. I think we've been looking a little more broadly at these models that just the proof of concept that this is actually making these changes. We don't really have any studies that sort of actually look deeper than things like collagen and inflammatory cytokines, at least right now. Yeah.
Why is ABC associated with ER stress?
So the first, the very first paper that kind of put ER stress on the map was in obese mice in 2004. It was a science paper. So no one really knows per se why ER stress occurs. Is it an increase simply in the folding load, right? Or is it a decrease in the folding capacity? There's certainly some evidence that the folding capacity is diminished. So some of those chaperones, for instance, can be decreased. If we look at it at this very simple level of just a balance between folding capacity and folding load, if that's the case, there's evidence that the folding capacity is changed. There's also evidence that I kind of really vaguely got into, that things like palmitate may be able to actually induce the EPR totally irrespective of protein load. So a palmitate may be able to actually stimulate the EPR on its own, and that's what's causing this EPR induction. But it's all speculation at this point. Yeah.
How permanent is the damage to the lining of the gut? So when you increase gut permeability, can you reverse that?
Mm-hmm. Yeah, certainly by this measure, by things like fits increasing. Yeah. Is there a hand up? Yeah.
I never understood this model. Maybe this is why your reviewers don't like antibiotics too, but I thought the antibiotics just wiped out all microbes.
Right, that's a good point. And so if you didn't catch that, so if antibiotics are wiping out all the microbes, then presumably they're actually wiping out some good microbes, right? So then why would it be totally protected? Again, we're speculating, we don't know for sure. Our thought is that at this point when they've been fed a Western diet, there's a shift such that their microbiome is predominantly now potentially pathogenic. So even though you may be wiping out some of the good microbes, it's predominantly bad ones that are wiping out. That's the best answer I have. Yeah.
So if you'd had a standard diet antibiotic group, you would have seen.
That would have been interesting, right? And that's why I mentioned we really should have. We got a whole bunch of grief from the reviewers that we didn't. There's some evidence from other groups that antibiotics in controls it doesn't do anything metabolically. But you certainly could, if I was correct in rationalizing why they're protective, then it might have a very different effect in control. It could be detrimental, right? Yeah.
So outside of ER stress, is there anything that might be happening to the endothelial cells that could be decreasing arterial compliance?
Outside of ER stress.
Right, so any other kind of cellular stress that might be affecting stiffness?
Stiffness, I'm sorry, and the stiffness in general?
Right.
Oh, yeah.
Specifically with the endothelial cells, though.
Oh, the sort of, the crosstalk with endothelial cells and stiffness? Mm-hmm. Yeah. There's a lot. Yeah, I think ER stress is just one stress pathway out of dozens that could be controlled, that could be affecting endothelial cells. There's yes, absolutely. Yeah, again, ER stress is pretty narrow. When we have these models of obesity and diabetes, there's so many different cellular stress pathways that are upregulated that have nothing to do with ER stress. Yeah.
I'm just curious about the Western diet versus the standard chow diet. So the chow diet was like 4% fat, I think, and the Western diet was 46 or 48%.
I think that's right, yeah.
How does that compare to a low fat versus a high fat diet in humans? Because that seems like a big percent.
Yeah, so
increase in percent fat.
Right, it's huge.
Yeah.
It's bigger. Another thing I can say is, we're only, in a sense, we're not looking at the dietary effects. We're just using the diet as a means to an end, right? We're just using the diet to cause obesity. So the low fat is really low fat, much lower than a human diet, right? 46% actually might be reasonable for a human. But again, we don't worry we do in some cases, but we're really just using that diet as a means to an end, just to try to reduce the dysfunction of obesity. So we don't worry too much about individual nutrients and stuff. But you're right, if you're to relate it to humans, the low fat, really low fat.
So a lot of these models, aging, obesity, probably diabetes and obesity, are characterized by elevated sympathetic nervous system activity. So whether it's circulating catecholamines or just neuronally released norepinephrine, is there any evidence to suggest that chronic elevations and alpha receptor stimulation can lead to ER stress?
No, I thought you were in a different direction. I've never seen anything linking ER stress to alpha receptor stimulation. There was a paper last year that came out in mice linking norepinephrine to changes in the gut microbiome. I think it was in hypertension last year. But I've never seen anything that are linked to ER stress. Yeah.
What did it show related to the microbiome?
I have to go back and look, but it was essentially norepinephrine, I'm pretty sure, being released from the nerve terminals, again acting kind of feedback to change the microbiome. But I'd have to dig it out to figure the details. Yeah.
So I just want to follow up on the diet question. So I recognize that the diet's a means to an end. It's the way to make the mice obese. Humans don't become obese overnight or even over a couple of weeks. Like it takes a long time to become obese. So if you're expanding the fat pads that quickly, are you creating additional stress?
I would actually argue this isn't that fast, right? I mean, if you look at the lifespan of a mouse, right, being 2.5 years, we gave a diet for 5 months. And I'd actually argue if you look at a percentage of their life, it's actually pretty long. And this is much longer than a lot of people are giving diets to mice for 8 weeks. So ours is pretty long. I mean, you're right. Yes, I think there is a change. If you expand those fat pads quickly, it probably does cause a different metabolic stress. And so there is, yes, likely a limitation. But I think in the scheme of looking at Western diets and mice, ours is actually pretty good.
All right. Thank you.
Thanks,
Dr.
everybody.
Gotta go get me some.
Yeah.